Our interest was piqued by the allelopathic qualities of black walnut trees. As a result, our research question was, what effect does allelopathy in black walnut trees have on the species richness of woody plants? According to McCoy et al. In 2022, many organisms use a tactic called interference competition to obtain resources by reducing the ability of another competing organism to obtain shared resources. A specific type of this competition is called allelopathy, which is used by plants and involves the secretion of chemicals to negatively impact the growth of other plants. Juglans nigra, commonly known as a black walnut tree, secretes a compound called juglone that impacts the germination, size, color, and lifespan of nearby plants, which is described by Donna and Lerner in their 2001 article and by Cesar in 2009. This allelochemical is most concentrated when secreted through the roots, though it is present in all parts of the tree. In our research, we discovered that there are some families of plants that are quite tolerant to the effects of juglone. Donna and Lerner in 2001 claimed that maples, the rose family, beeches, and oaks can tolerate juglone. Furthermore, they explained that species within the same family as black walnuts, including pecans and hickories, are resistant to juglone. Due to the concentration of juglone in the roots, we have decided to focus on a measurement explained by the University of California, Berkeley, called the critical root zone, which can be defined as an area of ground surrounding a tree where most of the tree's roots reside beneath the canopy. We'll learn more about this measurement when we talk about our experimental methods. Given our research on this topic of allelopathy, we hypothesized that black walnut trees have a negative effect on the species richness of surrounding woody plants when compared to non-allelopathic tree species. To test our hypothesis, we decided to count the number of species of woody plants in the critical root zone of a black walnut tree. To calculate the critical root zone, we first had to measure the diameter of breast height, or dBH, of the tree. From there, we multiply the dBH by 0.12, which gives us a radius that we measure from the base of a tree. Using this radius, we create a circular zone around the tree and mark it with flags. Then, we count each of the woody species in the CRZ. We measured a few other items as well. When getting started, we noted the time, date, and weather, determined the tangent height and geographical location of each tree. Then, as we counted the individuals, we noted the species and measured the dBH of each woody individual in the CRZ. As with any experiment, we must establish a control group to compare our data to. Based on our research, we decided to use shagbark hickory, Caria ovata, as our control group. We chose the species of tree because it is in the same family as black walnuts. According to Donna and Lerner in 2001, shagbark hickories also produce juglone, but its toxicity is so low compared to black walnut that it is rarely observed to harm other plants. We primarily collected data at the Home Quarry Recreational Center, except one data point collected near Todd Lake. As part of the National Forest, these areas are minimally impacted by humans. Based on our research and experimental methods, we predicted that if a black walnut tree is present in an area, then fewer woody species would be present. Now let's take a look at our results. This first bar graph shows the average species richness, or number of species, found within the presence of a black walnut or shagbark hickory tree. The average number of species found within the critical root zone of the black walnuts is 6.2 species, while the average number of species found within the CRZ of shagbark hickories is 7.0. The next data analysis we performed was to determine the characteristics of the species within the critical root zones. To do this, we researched all of the found species to determine whether or not they are tolerant to juglone. As depicted in the bar graph, there were fewer species that were not tolerant to the chemical near black walnut trees than shagbark hickory trees. Since fewer species were found near black walnut trees, we can support our hypothesis that they negatively impact the species richness. However, these results are not likely significantly different. Because of this, we decided to inspect whether or not the species richness of those that are known to be intolerant to juglone will be impacted. This data collected suggests that juglone-intolerant woody species are not as present around black walnut trees. When collecting data, we encountered a few issues. First, this project was started in mid-October. As a result, many of the trees were turning color and losing their leaves, which made it difficult to identify the species. Another issue we had in collecting data was locating the trees of interest. While we had researched the general habitats for these trees prefer, we still had trouble locating these trees because we were often just hiking the trails of the National Forest with little to no direction. Overall, while our data did support our hypothesis, the data did not seem strong. As a result, we would recommend repeating this field study to gain more data points and perform a proper statistical analysis to determine significance. If further research is to be done, we would also start earlier in the year, perhaps in late spring or summer, so that identification is simpler.